The survival of humans as a species has and will always be dependent on external sources, like plants and animals.
Plants are the basis of most natural ecosystems, as they provide us with food, medicine, and clothing.
The agriculture industry is one of Canada's top 5 and accounts for over 8.5% of its GDP. Canada exports about $24 billion worth of goods every year to over 180 countries around the world.
So it isn't a surprise that growing plants is the livelihood of many people. But in some areas, what's needed to grow themselves is lacking. Water is not always abundant, and farmers sometimes have to cultivate plants in regions with extreme environments or soil with minimal nutrients.
The purpose of this lab investigation was to explore the effect salt would pose on plants and crop growth so that farmers can better understand the phenomenon that is occurring to their plants.
Salt, using a process known as osmosis, draws water out of microbial cells through a semi-permeable membrane.
This process of osmosis, which commonly occurs when a higher concentration of salt is outside the cell membrane than inside, is expected to cause the plants to wither through low turgor pressure. Low turgor pressure takes place when water inside the cellulose-made cell membrane is not in enough sufficiency to originally hold it erect.
Six 330 milliliter beer bottles. One by 60 centimeter balsa wood. 6 pieces of 20 centimeter string. 6 pieces of 10 by 1.5 by 1 centimeter celery.
Firstly, in order to have 10, 25, and 50% of salt-saturated solutions, we need to calculate the amount of salt we need for each independent variable for 300 milliliters of water. Since 35 grams of salt is the saturation point for 100 milliliters of water, that means 3 times 35 grams of salt is the saturation point for 300 milliliters of water, which would be 105 grams. Secondly, we calculated 10, 25, and 50% of 105 grams. 10% of 105 is 10.5 grams. 25% of 105 is 26.2 grams. And lastly, 50% of 105 grams is 52.5 grams. Then, stir the salt into 300 milliliters of water, creating different salt solution using the magnetic stir. Meanwhile, the containers with 300 milliliters of pure water and no water are the control and negative control variables. Using a funnel for rate and ease, pour the salt solution and water into different bottles at the same time. After, measure the mass and insert the pieces of celery. Watch for qualitative data. As for quantitative data, measure the mass of the celery pieces after 12 hours, which would be your dependent variable.
This graph shows the mass differences of the pieces of celery before and after the experiment for the 5 solutions shown in the graph. The y-axis shows the mass measurements in grams for the tests. As you can see in the graph, before the experiment started, all celery pieces were 20.9 grams, but after the 24-hour occurrence, only the mass for which the celery was put in the water increased, while the celery mass in no water, 10, 25, and 50% salt solutions decreased by 1.7 grams to 3.7 grams. Some qualitative observations that we gathered are that the celery with no water dries up and turns yellow. This is due to dehydration. As the celery in salt water withers through low turgor pressure due to osmosis, it weakens the celery, thus decreasing its mass.
Based on the experiments that we have conducted, we can base the conclusions that was expected. Any added salt saturation doesn't aid the growing process of plants, and actually has a negative effect on plants due to the process of osmosis. As the water is drowned out of the microbial plant cells through the cell wall in between the membranes, less water is provided to the plant to provide nutrients required for growth and development. If our experiment was set up so the celery was sticking out of the solution, you could also observe the low turgor pressure. You would see this as the celery would progressively become less erect and limper. From our experiments and theoretical knowledge combined, we can see that additional salt percentages will harm and in no way benefit the plant at hand. The next step for the scientific research would be firstly to warn farmers to avoid any objects containing salt contents to be in waters as they water their crops. Secondly, to experiment with other solutions than only salt and water, then compare with the negative and control baseline data from this experiment to discover a perfect solution that will help farmers around the world.